Hello, my name is Professor Matthew Schmidt, and welcome back to Genetics. We're going to continue our discussion of DNA structure, and in particular in this session, we're going to talk about DNA replication. We started to talk about it a little bit conceptually when we finished up last time, in the sense that when Watson and Crick came up with their double helical model of the DNA molecule, right away they saw they couldn't verify it experimentally, but they saw a way that DNA could replicate itself. And we'll talk about that in a minute, but I was thinking about this earlier. When we say that DNA replicates itself, this could be potentially a little bit misleading. It's not as if you have a DNA molecule and then just all of a sudden it alone makes a copy of itself. What that term means, and you'll hear it a lot, is that DNA possesses the information in the correct way that it is able to be copied so that two full new copies of the molecule result. As we're going to see, there is a ton of machinery that's sort of necessary for this process to work. So DNA copying itself sort of means informationally, not really, it's responsible for the process alone. Now, there's a lot on this slide, but I wanted to connect it with Watson and Crick's model of DNA structure. And remember, structure and function are always so intertwined, and really you can't take them apart. So we had noted that one of the requirements for the genetic material was that it had to be able to replicate itself in the sense that we've said. And, even Mendel could have told you that, because he said that, perhaps more the chromosome people, but either way, it was known for a long time before we got to this point that when a cell copies itself, both of the two new cells have, a full complement of chromosomes, for example. So somehow the DNA has to be replicating itself. And remember that Watson and Crick did realize that their model could provide a relatively easy conceptual explanation for how DNA replicates. We saw this, and I'll show you a picture again in a minute, but basically the idea was that, before I read what I have on the slide here, just think of it. We said this, I think, before, and we'll say it again. If you know the base sequence of one side of one of the strands of the double helix, is it not true that you automatically know the base sequence of the other strand? It is true because of those base pairing rules, right? That A always goes with T and G always goes with C. So that's really the essence of this. So they said that they could envision, as I'm sure you can, the double helix sort of being unwound or unzipped, and that each single strand now, them being separated from one another, would act as a template to construct a new strand, because and solely relying on the base pairing rules. And that would create two new, at least informationally identical DNA molecules. Now, a term that we haven't talked about before is that they noted that if this is the way that it happens, that this replication process would be called semi-conservative or semi-conservative replication. Because, as we're going to see, in this model, half of each newly replicated molecule consists of the old or original material, and half of the material is new or newly synthesized. So let's look at this in a little bit more detail, and we'll see that while it's great that Watson and Crick said it, experiments had to be carried out in order to verify that in fact this was what was going on. First, I just wanted to show you the basic picture. I think we might have seen it last time, but if we didn't, we talked about the idea. So this is a just conceptual simplified model based on the double helical structure of DNA, showing how Watson and Crick and perhaps others envisioned that it could be replicating itself. So above here you see sort of the traditional double helix, and note that the sugar phosphate backbones, which are drawn as ribbons here, they're drawn in purple, and there's a reason for that. We want to be able to differentiate the old from the new in the sense of semi-conservative. So right here is where the replication is actually happening, but what we want you to see is that, let's follow this purple one down. I'm doing it in yellow. These two strands that I'm now copying in yellow, they were part of that original double helix. and they've unwound. Well, this drawing is supposed to be sort of a composite of the process. We do have an animation that I definitely suggest you look at that shows it unfolding in real time, and I mean both unfolding in time and literally unfolding the molecule as well. But you know, a video, if a picture's worth 1,000 words, an animation is worth at least 1,000,000. But I want you to understand here what's going on. So if those two strands came apart like that, now they're free, and new complementary bases are going to almost automatically join in. In other words, here's the reason. Look at this area here where we're putting our attention. So you could look at it from either side of the helix, but if there's a C in this position here, and there's a G in this position there, once it gets opened up, well, what's going to happen? See, these two parts down here have already been replicated. So, but right here we're seeing, for example, here's an A, so we see a T coming in to join it there, right? Here's a T, we see an A coming in to join it there. So this is a process that's going on. And when you're done, notice that this whole molecule and this whole molecule, which eventually will become separate, are informationally identical. In other words, you have a CG, AT here, you have the same thing over there, and if you look at every single base pair in both of those two new molecules, they're going to be identical. So this is the key conceptually to understanding how DNA has the power, if you will, to replicate itself. Now, as far as the semi-conservative nature of this replication goes, look at this. In red, I'm going to go over, they're sort of in pink here, but I don't know if the drawing really gets it across. These are the newly made strands over here that I'm drawing in red. See, it's sort of, I hope this is making sense. Up here, there's no newly made strands in this area. Here is where the new nucleotides are coming in, and now we have two new molecules. But now you can see the idea of semi-conservative. So if this one here, once it becomes its own whole full new molecule, one half of it, that which is written in yellow here, is literally the same strand that existed up, I don't want to use that color, sorry, that existed, you know, up here that we were copying from before. It's literally the same. It unfolded, it unzipped, and it served as a template, but it's part of that new molecule. What's written in red over here represents the newly incorporated bases. So the semi-conservative idea says we get the split, each of the original strands acts as a template, and two new strands are built. Now, even though they're part old and part new, I keep saying informationally. In other words, if you forget about the red and the yellow, if you just look at the nucleotides and the sequence information, they are literally identical in both of those two new molecules, as they must be, right? So if we think of each of these two new molecules as daughter molecules, we have the old strand, which is written in purple on the diagram that I wrote in yellow, and the new strand, which is sort of pinkish on the diagram that I covered over in red. We see how this works, and it's very symmetrical and almost mirror image like. Not perfect analogy, but that's sort of what's going on. And what's happening is, at the risk of too many things being drawn in here, replication is proceeding in this direction. In other words, this part's going to get unzipped, and the same thing is going to happen as we go up until finally the whole thing has been copied and we have two full new identical molecules. Right? That's what Watson and Crick said. At least it was one of the reasons why their model was so widely accepted, and people said, yeah, that really makes sense. I mean, you may take it for granted. You may have been hearing for a very long time that DNA is a double helix and figure that's just the way it is. But it was not known, obviously, until they published this in, I believe, 1956. But being scientific, it's all well and good, and I mean that seriously, to propose a hypothesis about how DNA replicates itself. But Watson and Crick were not experimental scientists, really. Crick went on to be one, but I mean, when they were doing their DNA work, a lot of it was just thinking about what was known and trying to figure out how it could all make sense. So the experiments that actually I guess I'll ruin it by telling you that pretty much proved, that's a strong word, but they lent very, very strong support to the fact that Watson and Crick were right in terms of the semi-conservative nature of DNA replication, were carried out by, now I didn't want to do that, I'm sorry, and I didn't want to do that either. I wanted to do this. Sorry about that. The experiments were carried out by Maselson and Stahl. These are famous experiments that I can almost guarantee you're going to need to know about. So they said, okay, Watson and Crick and whoever else said this, how do we really find out if that is what in fact is happening? And I've seen a lot of test questions about this, so it does pay to understand both what they did and how the data can be interpreted. So they said, look, on the one hand, hopefully you understand the general idea of semi-conservative behavior of this right now, but there were other possibilities. In other words, if there was a conservative model, and they totally considered that this could be the fact of the matter, somehow each entire double helix acted as a template for the creation of a completely new double helix. So if we did that based on colors, what we'd be saying is, I'm not able to draw a double helix in a really expert fashion, but semi-conservative, we're basically saying that you start off, we're using colors to keep the old, right? Basically what happened, guys, think of it this way. The two strands separated, and then I'm going to draw in yellow, well, maybe red is better for here, two new strands. It's not the best drawing, but you get the idea that the two new molecules are, each of them has the half that's the original template, and half that's newly made material. If it was the conservative molecule, the conservative model, excuse me, I'll write that over here. You're starting with the same molecule, right? But somehow that entire molecule is going to serve as a template to make an entire new one, meaning that when you're done, you're going to have actually the original molecule still in existence, plus a completely new one. And while you may say, well, why would they think that? They're just saying that that's another way that this could happen. We don't know. That's why we're going to do the experiments. Now, there also was another possibility called the dispersive model that I don't want to get too in-depth into. You may need to understand it, but really we're going to focus on semi-conservative sort of versus conservative, and you can take the rest from there, and in the problem solving, certainly you can do some problems about that. So here's the rationale, sort of the logic behind their experiment. First thing they did, and the experimental rationale is not the same, but we've seen this type of strategy before in that they grew E. coli, typical bacteria, on so-called heavy nitrogen. Now, it happens to be radioactive, but for this experiment, that really is not important. It's heavy in that it's 15N, normal nitrogen, if you will. This is an isotope that's heavier. Normal nitrogen has got a weight of 14. So if you grow the bacteria on heavy nitrogen, it's going to incorporate that heavy nitrogen into all of the molecules that that bacterium produces that have nitrogen in them. That includes proteins, that includes DNA. Remember, we're not at the point now where we're trying to distinguish protein from DNA. The bottom line here is that they're going to isolate the DNA, and all the DNA after growing the bacteria on this media for a while is going to contain heavy nitrogen, okay? So they grow it and grow it and grow it. All of the DNA's got heavy nitrogen in it, which makes the entire DNA molecule literally heavier. I mean, it's not like it's, at the molecular level, it's heavier, and there are ways to figure out which are heavy and which are not. So they have these bacterial cells that contain DNA that's all heavy. After a time, and I don't specify because it really doesn't matter, it's just they grow it long enough to be pretty clear that all the DNA is labeled heavy. When they were sure it was all labeled, then they collected that bacteria and they switched it back to a plate that contained normal 14N, non-heavy nitrogen. Now, what would happen is each cell division cycle in bacteria takes approximately a half hour. So they wanted to see, okay, after one generation, what that really means is since when bacteria divide, they do binary fission, that DNA molecule has copied itself and now is ending up in both of those cells. So they said if we look at the DNA from cells after one generation, which means after one division or copying cycle, after 2 generations, after 3, after 4, we want to see what kind of DNA is in there in the sense of, is it heavy, is it light, is it a mixture between the two, etc. Now, how do you figure out what's heavy DNA and what's light DNA? This was in the 1960s, I believe. They really didn't have technology like we have today. But what they could do was centrifuge the DNA in a so-called cesium chloride gradient. As far as I'm concerned, you probably don't need to know that much about a cesium chloride gradient. It's a salt, and what it is it creates a density gradient. And sometimes you centrifuge things in this for over a day. The bottom line is that molecules of different densities will find their place, and they'll stop moving when they're in the gradient that corresponds with their density. So it's a way of separating out molecules basically that are different weights or densities. So you'd end up having different bands in the cesium chloride gradient depending on if a molecule was heavy. The heavier it was, the further down it would go. So based on predictions, which we're going to get into in a minute, when they looked at their data, they determined that in fact DNA does replicate semi-conservatively. In other words, Watson and Crick were right. But you're often asked to sort of either interpret or analyze this data, so let's check out what exactly they did and what they saw. So we have up here DNA being spun down in a cesium chloride gradient. That's just sort of a depiction of the experimental equipment. But here's the idea. So what they said was, here are the possible results. First, we have to get this in our mind. So before they went and did this, they said, all right, let's just take DNA we know is heavy. How do we know? Because we grew the bacteria for, however long on this heavy nitrogen, so we know that the DNA is all heavy now. And they got sort of a marker, sort of a reference. In other words, at this spot, they knew that DNA that contained all 15N, all heavy nitrogen, would get spun down to this area right here. Then they could also do it if it was just 14N, right? In other words, grow the cells on normal nitrogen, extract their DNA, spin it down, and that ended up over here. So clearly you could separate them, right? And if you had a hybrid between 14 and 15N, that would end up somewhere in the middle. So with this known, they could then go ahead and isolate DNA from their different bacterial generations, see where it ends up, and see what conclusions could be drawn. So let's check that out. So in step 1, as it were, of the Maselson and Stahl experiment, they, as we just said, they grew it on the heavy nitrogen, collected some DNA, always on the heavy nitrogen, spun it down, and they said, there it is. That's the position where fully heavy DNA is going to end up. Then they allowed the bacteria, they transferred the bacteria to the 14N containing media. They let them go through a replication cycle, and then they wanted to see what happened, right? And this is the really important part. So step two meaning after one division in non-heavy nitrogen, let's think about the predictions conceptually. If the semi-conservative model is true, let me draw it in yellow because we have a black background here, then the way they did it, all of this, think of all this was heavy, right? Yellow is going to mean heavy now because that's what they grew it on. Now, if semi-conservative model is true, the strands separate, right? But now the light nitrogen is going to be incorporated when the DNA goes to copy itself. Let's draw that in red. So just like we did before, we see, sorry I can't draw helices too well, we're going to see half and half. And doesn't that mean that if the yellow is heavy and the red is 14N light, these molecules are going to have a density right in the middle between 14 and 15, right? It's a little odd to say 14 and a half, but that's the idea, because each of those molecules is comprised of one 15N strand and one 14N strand. So if the semi-conservative model was true, all of the daughter molecules that came from after one generation of bacterial growth should be hybrids in terms of weight, right? And that would mean that all of them should go at the point that was somewhere, I didn't draw it on here, between the pre-established 14N and 15N locations. And that is exactly what they saw, okay? It's important to realize that if the conservative model was true, let's draw that over here. So you'd have all heavy. My drawing skills are even deteriorating. And we had said if the conservative model was true, somehow the whole one molecule in the next generation would be original. But see how their genius may be a little much, but it was a very clever experiment, because if the conservative model was true, can you see that you'd have something that was completely heavy there and something that was completely light? So if the conservative model had been true, and I think the way I animated this, it may erase something, but if it was conservative, oh, it didn't. So you'd basically see, I almost got them right. I'm very proud of myself in terms of position. If conservative, I'll write in green over here, was true, you would have seen one band down here, because that corresponds to that heavy molecule, right? And one band up here. So even after the first generation, you pretty much can tell that if it was conservative replication, you would have seen these two bands. If it was semi-conservative, you would have seen this one band. And in fact, you saw that one band. So right there, that's strong, strong evidence that semi-conservative replication is what's happening. And I just drew these two molecules over here. I didn't draw them, but to show you that it's sort of a blue and a purple, showing you that each of the new molecules is in fact a hybrid. I don't want you to confuse the information. Informationally, these are equal to each other, right? But physically, each of those molecules is comprised of one original older strand and one newly made strand. And this experiment is the proof that that's the case. All right? Now, sometimes just because they want to, they'll say, well, if the conservative model was true, what would happen after 2 generations? Or if the semi-conservative model is true, what's going to happen after 2, 3, 4 generations? We're not going to take it ridiculously far right now, but we could think about those questions. So step 3 simply means they looked at the next generation. And let's, before we even look at the results, let's just look at the predictions based on. So now, if it's semi-conservative, we're taking molecules that would be like this, right, from last time, after the first generation. I almost want to say F1, but you really shouldn't use that terminology here, right? But it's the idea, the offspring. They're going to now divide. And look what should happen. What's going to happen? So these two strands will separate, right? We got to keep track of each one of them. But now the whole thing is growing on light nitrogen, which we made our red color. So all the new DNA that's coming into this situation is going to be light. So what would you see if in the second generation, or after 2 copy cycles, if you will, if semi-conservative replication was true? Well, you'd see 2 strands that were hybrid 14-15N, just like you saw before, but now you're also going to be seeing 2 completely light new strands, because that's the, the nitrogen that's available to make DNA now. So I said that we weren't going to write it, but it was already written here. If semi-conservative replication is true, you're going to see some light DNA, which represents these molecules we drew in red, and some hybrid DNA, which represents these molecules here and here that are half and half, right? But again, and these molecules over here are just showing you that, I guess the purple color here, is equivalent to our red, and the more blue color is equivalent to our yellow. So you're seeing 2 of them, all brand new, right? They're going to be the ones that end up here, and 2 of them sort of half and half Those are the hybrids, right? Now, what if it was conservative, you guys? It's important that you understand this. If it was conservative, remember what we said would have been happening last time. We had two molecules. One was totally light and one was totally heavy, right? Well, I'm sorry. That's true, but I reversed them. This was the light. Darn it, I'm sorry. This was the heavy because they started off on the 15N, and this was the light. Now, if we follow this logic, what's going to happen? Each of these molecules is now going to serve as a template. I'm going to have to write a line all the way down here. Now, all that's the same idea. We only got 14N around now. So this one is going to stay like that, but its new molecule is going to be all red. And this original red one is going to stay there, and the new one is going to be all red. So again, you're going to see one band that's completely heavy and one band that's completely light. And in fact, you'll see more and more actual material in the light area as time goes by. So I think I animated it correctly again. If it was conservative, we'd see a band here that represents this, right? That's all heavy. And we'd see a band up there that's all light, which represents this. And in fact, with the conservative model, sometimes they'll show you that there'll be more material at that level. So if they ask you just where are the bands, that's one thing. If they ask you how much material will be there, in the semi-conservative model here, the same amount would be at each spot. If the conservative model was true, there'd be 3 times as much DNA up in the light area as there was in the heavy because of this. And I hope you can see that. You can go on and on and on with this, but I don't want to, and I don't think you want to either. I'm joking, obviously, but I mean, we've already spent a good deal of time on this, and in these sessions, the most important thing is that you understand what's going on, okay? And while I look down at my podium here, it looks like a complete mess at this point, but if you're following along, you should at this point understand both the conceptual rationale for semi-conservative replication and the Maselson-Stahl experiment, which led to the actual evidence that that was in fact what was going on. And the alternatives, you should be aware of what would have happened if the alternatives were true, okay? Because often that's asked. But you may wish to take a break now because we're going on to a topic that's related. The reason I put them together in a rather longish video is because often they're considered in the same textbook chapter or sometimes in the same lecture. It may or may not be. So part 1 is the Maselson-Stahl experiment and the rationale. Part 2 is what's going on at the molecular level. Because it's all well and good, and again, I don't mean it sarcastically, for Watson and Crick to propose this idea and for Maselson and Stahl to confirm it. But that's a far, far cry from really understanding what's going on at the molecular level. Remember, we said DNA just can't up and say, I'm copying myself. It has the information to do so, but it turns out that it needs a lot of help. And at the molecular level, it's actually rather complex. I'm not even going to go into all of it, but I'm going to go into enough that you understand the basic idea, and almost always you do have to know this. It turns out, I'm not sure if we ever actually said this before, that almost always a chromosome is one long single DNA molecule. The length depends on what chromosome it is. And it contains at least one, usually more, but it depends on the situation, area that's called an origin of replication. That's a base sequence where the beginning of replication can occur. Now, when that's initiated, the replication proceeds in a bidirectional fashion, creating 2 replication forks. So if we, my drawing scares me a little bit, but here's the idea. Just think of a DNA double helix. The origin of replication is right around there. And what's happened there is that unwinding has occurred, and we have almost what looks like a bubble there, and then whatever color we want to draw, those areas are now acting as a template to try and make some new material. And the replication fork is right here. They're proceeding outward in each direction from the origin of replication, so that we're going to see later what the consequences might be, if depending if the DNA molecule is circular or linear. But right now, you're basically seeing that once it starts, it's going off in both ways. In order for this to be successful, there are a lot of enzymes that are involved in a process that is fairly complex. So we're going to start with the major ones, and DNA polymerase is the vital and necessary important enzyme. That's the one, it has actually a couple of functions, but it's one thing to say that new nucleotides sort of automatically come in. There is a natural affinity, a chemical affinity between A and T, for example. That's all well and good. Let me just draw a little picture over here. Say you had a sequence of A, G, C, T, right? And this strand was open to be a template. And new ones are coming in based on the base pairing rules, right? But I don't know if you ever thought of it, but they're just sort of sitting there. What we need, and what DNA polymerase does, is an enzyme that polymerizes the actual making of the covalent bond that holds this strand together, so that we really have a strand that's properly held together with covalent bonds. All right, so that's really what one of the main things that DNA polymerase does. This may or may not be gone over in your particular class. What we call DNA polymerase is actually a rather complex assembly itself of many polypeptides. So it's a multimer, if you will, type protein, as many of them are. The whole thing is called the holoenzyme, made up of various parts, okay? But so the main thing here is that we need to understand why we need certain enzymes and what their names and functions are. So clearly we need an enzyme that's going to, it doesn't really need to align them so much, but if you want to think of it this way, is DNA polymerase is responsible for saying, okay, there's an A over there, a T goes here, there's a G, we're going to put a C in, and we're going to catalyze the formation of a covalent phosphodiester bond between those two new nucleotides. Otherwise, the whole thing would not really be stable. Now, one very interesting thing about DNA polymerase, and one that confounded scientists for a long time, is this. It can only create a new strand of DNA in the 5' to 3' direction. This is so important that we need to take a minute to understand what this means. We'll see a much more detailed chemical picture in a moment, but the bottom line is this. You guys know, I'll try and make it as simple and as good a drawing as possible, that we have the double helix like this. Because it's anti-parallel, which we discussed, if this red strand is running 5' to 3' this way, this blue strand is running 5' to 3' that way. All right? It turns out that templates are always read in the 3 to 5 direction, which means in yellow, yellow doesn't show up on white, sorry, green. A new strand is being made off of that blue template in that direction, in the 5' to 3' direction. That's the way it has to grow. It could never on the red strand be going, it could never on the blue strand be growing that way. On the red strand, it would be growing that way. And this causes a problem, because we're going to see it in a moment, but if you just unzip the DNA and you have these two open template strands, one is going to be going in this direction like this, and one is going to be going in that direction like that, right? Well, what this means is that one strand can just merrily go along its way, one new strand being produced, but it almost seems like the other one over here has to go in the backwards direction. If the replication fork is moving like this, one strand seems to have a big problem. So for the longest time, people were looking for a DNA polymerase that could polymerize sort of in the backwards direction, making a strand extend in the 3' to 5' direction. Well, the bottom line is, they never found one, and it doesn't happen, and we now understand how this sort of strangeness can be taken care of. Before we mention that, though, I also want to tell you that DNA polymerase has a 3' to 5' so-called exonuclease activity. It's an error correction mechanism, you guys. This is a very large enzyme, we said, made of lots of polypeptides, right? So as it's going along polymerizing, the front end is doing that. Think of it this way. The back end is sort of trailing, and it's like making sure that the fidelity is correct. So it's possible mistakes get made, guys. The second law of thermodynamics, implies and almost mandates that things can't be perfect in terms of fidelity here. Mistakes get made, and that's just the way that it is. But we don't want mistakes to get made if we're DNA molecules. We want to copy with high fidelity. So if the trailing end, the exonuclease part of the enzyme, detects a mismatch, in other words, it says, okay, there's an A over here, there should be a T over here. What the heck? There's a G there. That's a problem. It basically says stop, hold everything. And it goes back and cuts that out. That's why it's going back 3' to 5'. Then it says, okay, I've gotten rid of the offensive, if you will, incorrect base. Now resume, go back ahead, put the correct one in, and I'll keep checking. All right? So the polymerization occurs in the 5 to 3, but it can go back to excise or cut out a faulty or incorrect, or maybe a better word even is, it's escaping me, but nucleotides are complementary. So if it detects a non-complementary base pair, that's a no-no and has to get rid of it. So it does create the theoretical problem, and I've sort of already said it and drawn it out, that since a replication fork proceeds in one direction, one strand, this is very important, is replicated continuously in our little drawing. That would be this one over here, right? It just chugs along. The other one is discontinuous. The bottom line being, let me erase this for 1 second if we want to keep our drawing, since it actually has to be polymerized in this direction, it has to wait for the replication fork to open up that area, and it's going to be made in small pieces that are subsequently going to be joined together. Thus, it is discontinuously synthesized. Sometimes people call this, often in fact, the leading strand, and people call this the lagging strand, because it literally does lag behind. It takes a little more time to get this all together if you look at the DNA under an electron microscope. Okay? Now, this was seen as rather strange at the time, and it was proven by the discovery of so-called Okazaki fragments. A Japanese scientist named Dr. Okazaki discovered this, so the fragments got named after him. And basically, during the replication process, he was able to isolate short pieces of DNA, which correspond to these over here, sort of like partially made pieces of the strand. And now there's another enzyme that we're going to have to bring into the picture, because clearly they can't stay that way. They have to be joined together. Ultimately, this all has to be put together into one big continuous covalently bonded strand, and DNA ligase is the enzyme that's responsible for that. So far we have polymerase that's responsible for making the strand and error checking, and we have ligase for, if you will, gluing together parts that have been discontinuously made that need to be put together. Now, here's the more detailed chemical picture that I wanted to show you, because I think it's really important and it's a potential place where you could get confused. Here is a deoxyribonucleotide, right? And the base is attached to the 1' carbon. And we have said before that the bond polymerizes through the 5', well, actually the phosphate group attached to the 5' carbon, and the OH attached to the 3' carbon, right? So what this has meant, we've tried to do it with this sort of effect. Here comes a new nucleotide that's going to be joining into the chain. This explains why it can only grow in the 5 to 3 direction. So this new nucleotide, see the way this arrow is drawn is meant to, this thing is like coming in. We're not drawing every single atom there, but basically a bond is going to form between phosphorus here and an oxygen To be honest with you, I forget the exact chemistry of it, but it doesn't matter for this. The bottom line is that the attachment is going to go here from the 5' phosphate on one nucleotide incoming to the 3' OH group that's on that carbon there, right? What if you just think of this as one little dinucleotide, two of them together, it's going to have a 5' end and it's going to have a 3' end, right? And if another nucleotide's coming in down here, that's always going to be added to the 3' end. So if you think of it in a different way, this thing is growing this way. More are going to get added on, and they'll always be added on to the free 3' end. All right? So it's rather important to understand that. That explains that's what DNA polymerase does, at least in its capacity as a polymerase. Always adding, in other words, there'd be no way to add it the other way. We couldn't take a nucleotide and stick it onto the top. It's just not the way the enzyme does it. Now that's one isolated instance. We've tried to show you here in a larger scale sort of strand type of an idea that if this is the template strand, this could seem confusing. I mean, I'm saying that the new strand is growing in the 5' to 3' direction. This arrow looks like it's going the opposite way, but what's happening is this nucleotide is flying in, and the bond's being made between that 5' phosphate, the 3' end there. So it is growing in this direction because new nucleotides are coming in from that end over there, right? And you'll notice also the base pairing rules are guiding this to an extent. The DNA polymerase must be necessary to form this covalent bond here. Each of these is a phosphodiester bond, okay? So that is the only way that it can occur. Now, if we look at a little bit of the bigger picture of this, a lot of that's great if you understand that so far, but unfortunately it's not as simple as that. DNA polymerase is a wonderful, great enzyme, but it's limited in a certain sense. And that is, not only does it have to only go in one direction, it does not know how to initiate, to start off a chain. It only can add nucleotides to the 3' end of an already existing or established chain. It's one of the reasons I just showed you that. So every chain must be initiated by the laying down of a short RNA primer. Now this may just seem kooky. What's RNA doing in the middle of this? We haven't even talked about RNA that much, but here's the quick bottom line, just to take a little side trip. The enzyme RNA polymerase, which makes RNA from a DNA template during transcription, which we'll discuss later, it does know how to start. Just go right down there and start a chain and start polymerizing. The enzyme DNA primase, which is responsible for laying down an RNA primer during DNA replication, is a cousin. It's related evolutionarily to RNA polymerase. It's almost as if during the course of evolution, we know that RNA preceded DNA evolutionarily. So during the course of evolution, it's almost as if the cell said, look, there's already an enzyme around that knows how to initiate a chain. It just happens to do it with RNA. I'm not going to bother learning that skill. Let's let RNA start off the chain, and then we can get the DNA, which I'm going to draw in blue, and add that on and keep going. So every DNA chain that's growing has to be primed or started off via DNA primase, and primase lays down a little short piece of RNA. Okay, so now we're adding another enzyme to our arsenal. Plus, I hope you're thinking this, that RNA can't stay there. It's got to be taken away later. But the fact that it exists, I could talk a lot about the philosophical implications of it. It definitely implies that RNA was much more ancient than DNA. All right, so subsequently, there's a DNA polymerase. Again, I don't know if you're going to have to worry about this or not, but it's too much for this lecture. In bacteria, DNA polymerase 1 comes over and uses its exonuclease activity to remove the RNA primer and then go ahead and fill it back in with DNA, because there's always something to hook onto as these Okazaki fragments are being made. So more enzymes. I know this isn't enough for you. Enzymes called helicases are needed. The helix doesn't just fall apart by itself. In fact, most of the time it's rather stable. A helicase is an unwindase. It's unwinding the helix type enzyme. The other enzymes can't do their work unless this thing is opened up, and it doesn't just happen spontaneously. In addition, there are SSBs, single-stranded binding proteins, that are also needed. They associate with the unwound healing to stop it from going back together, from reannealing. After all, DNA wants to, if you will, wants to be in the double helical conformation. So if the SSBs bind to it, it blocks them from reannealing while every enzyme can get its work done. So what do we have so far? DNA polymerase, DNA ligase, we have DNA primase, helicases, and single-stranded binding proteins, which aren't technically enzymes, but all these molecules are proteins. Okay, now I hope that's not go back and make sure you understand all the players. This diagram is awesome. It doesn't allow you to go into the most technical details, but it really takes into account everything that we just said. So let's take a little time to look at it and make sure you understand this. When we say this is the direction of replication, that means the replication fork, which is right here, is moving in that direction. It's going to be unwinding the helix in that way, and the whole process is initiating down in that direction. Okay? What that means is, look at the polarities very closely. Obviously, this strand over here, the 5' end is here, the 3' end is there, right? That can be read correctly because this new strand here is being made in the 5', even though it's not written here, to 3' direction. That's the, quote, correct way. So even though here's DNA polymerase, it can just keep going, and as long as this helicase, which is going to unwind the helix, keeps unwinding, this thing can keep going, right? That's why that's the leading strand there. The lagging strand, on the other hand, has to wait for the helicase to open up this region of DNA, and then it makes a piece there. You see little pieces of DNA polymerase there. It opened up more, it made a little piece there. It's not in the diagram, but now it can make a little piece here. Now, what's not being shown on this picture is, I'll draw it in red, there's a little piece of RNA that starts every single one of these little Okazaki fragments. All right? In fact, it's not usually that, it's important that it has to happen. Even this starts with a little RNA primer. But the leading strand only needs one, and then it just takes off. The lagging strand has many, many, many RNA primers that eventually will have to be removed. Here comes DNA polymerase, says goodbye, goodbye, goodbye. And now I have something to hook on there. Sorry, I took away the whole thing. In other words, once this Okazaki fragment is made, I now have an area that I can add onto, and then ligase can come and literally join this all into one, continuous. I don't know if continuous is the right word since this is the discontinuous strand, but it's discontinuously made, but ultimately it has to all be put together into a stable covalent situation, right? And as this goes this way, you have the leading strand just motoring on along, the lagging strand being built piece by piece by piece and then glued together. It's extremely important to picture that. And again, I don't want to push it too hard, but I think you should watch the animation because it's really amazing to see it actually happening. Okay, now that's really the situation. There are more details that you can go into, but I think understanding it at that level is a good level to understand it at. It's mid-level. And often what you'll have to recognize is there is the mechanism is generally the same. Like so many things we're going to see between bacteria and eukaryotes, there are minor differences. And when I say minor, they can be significant and important, but the idea is always the same. So in bacteria, for example, the main reason there's a difference is that bacterial chromosomes tend to be circular, okay? And while that diagram that we just said can apply whether you're circular or linear, we're going to see that there's a little difference. So let's say here's a bacterial chromosome, right? Here's an origin of replication. There's only one of them. One of the reasons is bacterial chromosomes are very, very small compared to eukaryotic chromosomes. They may have, it doesn't sound small, but they may have a few million base pairs in them as opposed to billions. Okay? So what's going to happen is, remember what we said, when replication is stimulated or begun, the replication fork is going to move bidirectionally. Okay? And what happens is, right about in the middle, there's an appearance of a phase that looks like the Greek letter theta, which we've drawn right there. And what's happening is, because if you can picture it, the top, both of the strands are copying themselves, but they didn't get around to the bottom yet. So one of them sort of falls down, and it looks sort of like that, like the Greek looks like a smile, but theta has that appearance to it. I really don't know if you're going to have to know this. Part of this is still being worked out, but there are at least 5 DNA polymerases in bacteria. If you need to know, it's DNA polymerase 3. That's the main form that's responsible for the polymerization. That does the main part of the semi-conservative replication. One thing also that's in bacteria that you don't find in eukaryotes are enzymes called topoisomerases. That's a mouthful. And DNA gyrase is one of the most exciting and interesting of these enzymes. The reason these are needed, it's very hard for me to sort of make a diagram or a picture about this. But when something is circular, you guys, and you start to unwind the helix, this creates a tension inside of the molecule. It's not exactly perfect, but if you take like a little circular, I'm trying to see if I can get it up there, little circular piece of something, in essence, if you start twisting it, you're going to find that it introduces what in biology are called supercoils into this whole thing. There's no way to relieve the pressure. Well, the pressure has to be relieved, and that's what topoisomerases do. They realize that this pressure is going to build up, and they make strategic cuts that allow the chromosome not to become completely tense with tension in it. And that's necessary. And then when it's one replication fork, but when the two sides meet, you're done. The two circles separate, and you've made two new ones. So here's a great picture where in blue we have the original DNA over here. The new DNA is being drawn as red here. And this is what the one that looks like theta over here, because this top part has been replicated, but the forks are going this way and they're going this way. So at that point, you have two parts that are completely replicated on the top, one of which is sort of drooping over, and the bottom is waiting. And as it continues, you see that here the two forks are just about done. There's only this area here remains to be polymerized. When the two forks meet each other, the two new circles separate and replication is complete. I wanted to say it's as simple as that, but it's really not that simple. But this part isn't so bad. Now, in eukaryotes, the big difference is, well, there's two big differences. One eukaryotic chromosome may have, I can't say the exact number, maybe close to 1 billion nucleotides. The DNA polymerase speed, it's pretty speedy, but it's got its limits, and we need to replicate chromosomes quickly. So because eukaryotic chromosomes are long, they always contain multiple origins of replication, sometimes hundreds, depending on the size of the chromosome. So that results in the appearance of replication bubbles, sort of like we drew before. Meaning, say you have your DNA, I'm not even going to draw it as a helix, long pieces of DNA. You might have a replication start there, there, and there, and as they open up, you're going to see these bubbles. And as replication happens, it's still bidirectional. As these bubbles meet, they'll join. But you have to have this or it would take a long, long time. I forget the exact figure, but I think it was said that it would, I don't want to make it up, but it would take a very, very long time to copy a whole chromosome if you had one origin of replication that just chugged along down that thing. I'm sorry to keep saying this, but I really don't know if you're going to need to know. There are at least 7 DNA polymerases in eukaryotes. Several are present at each replication fork, and I don't think that we have the time, and you may not need to know going into all of them right now. But here's another problem. Technical issues with the fact that eukaryotic chromosomes are linear. They're not circular, right? Why should that matter? Well, we're going to find out, but At the ends, the very tips of eukaryotic chromosomes are these pieces of DNA that are not informational. In other words, they're not genes. They're called telomeres. Telo just means the end. Remember, telophase is the last phase. An enzyme called telomerase makes the telomeres and replenishes them. Okay, we'll see why in a minute. Because what's going to happen is, because of the non-circular nature of this, the ends of eukaryotic linear chromosomes are degraded. They actually lose material, lose bases during replication. So sticking a telomere on the end is sort of like a buffer zone. So as that gets degraded, you're not actually losing information. And then telomerase can come over and re-lengthen it again. All right. Now, here's a picture of using the same blue and red. So the blue is the original. You see these replication bubbles. What that means is each of these represents an origin of replication. And it's going bidirectionally from each one of these replication forks are spreading out in both directions. And this is a progress sort of thing. So they're going further and they're going further, and eventually they all meet, and so now you have this big old bubble. But this is happening all over the chromosome, and eventually they all meet, and then you get two new DNA molecules, right? So we're not going to go into the molecular details of telomerase, but there are a couple of things we should mention, which is, see, the whole reason the problem exists has to do with the RNA priming, and that is, think of this template, which is in blue here, right? And let's focus in. I just don't have a lot of room, so I'm going to write it right up here. This is the very, very end of the chromosome, right? So on the one hand, the leading strand's been chugging along. It's coming down here, and it's like, oh, that's the end. All right, great. Done. But what had to happen over here? In order for the Okazaki fragment to be made on the discontinuous strand, there has to be an RNA primer there. So here's the last Okazaki fragment. So you could think of bunches of them down here all getting joined. But even if an enzyme comes and takes away that RNA, how do you fill that in? DNA polymerase relies on, it can't exist, but there would have to be some other next fragment there that it could hook on its new bases to. It can't do that. So that information gets lost. And if you keep doing this over and over from both sides, the ends start shrinking and shrinking and shrinking over the generations of DNA replication. Now, just a quick observation that's interesting. The enzyme telomerase, as we've noted, can replenish these, can add on more. So in theory, you never have to worry about losing information. Something that's very interesting is it was two things have been determined. In adults, as opposed to children, and the older you get, it seems that telomerase activity either really, really slows down or shuts off altogether. Some people have, this is a very viable idea, have connected this with a possible reason for aging. That, after a while, the chromosomes lose their integrity, the telomeres are non-existent, you start losing information. So people say, well, can't we just turn the telomeres back on? But it's interesting, but here's the thing about that. Sort of the opposite of cells not being able to divide are cells that can be immortal and divide forever. And that's what cancer cells can do in many cases. It was discovered a while ago that vigorous cancer cells have their telomerase activity up, up, up. So it is clear that you need telomeres if you want to replicate over and over and over and over again. The balance, though, between, so cancer cells, if we wanted to treat that, we would, this is just an idea, you'd want to get rid of the telomerase activity, right? So that these cells would not be able to proliferate so much. So without going into a long story, even without disease, there's always this balance between telling cells no, don't proliferate so much, and stimulating them to do so when they need to do so. Telomerase is just one part of that problem, but the existence of telomeres, if there was no RNA priming, there wouldn't be a problem. So that really shows how important that is. So at the end of these, only happens with linear chromosomes. Bacteria don't have telomeres. Where'd they put them anyway? Because it's a circle, right? You don't have this problem when it's just joining around, even with the RNA priming. If you think about it, it doesn't matter. Well, I honestly don't know how long this video's been, but it really feels like a long time since we began. So I hope that it makes sense to you, of course. I hope that you can take a little bit of a break and sort of let it sink in. And I also hope that I'll see you again real soon in another genetics session.